The nervous system is the system that allows our body to communicate with the different parts. The function of the nervous system includes monitoring changes inside and outside of the body. We call these changes stimuli. This sensory input is then processed and interpreted. We integrate the input. We make decisions based on that information. And then we have some sort of response via motor output. This activates a muscle or a gland. The nervous system can be kind of complicated, so let's take a little overview of what we're going to look at as we go through this system. We'll start with the tissue. There are basically two cell types in the nervous tissue: the neurons, which are the cells in charge of electrical signaling, and the neuroglia, which are the supporting cells. Then we will look at the central nervous system. The central nervous system is composed of the brain and the spinal cord. And finally, we will look at the peripheral nervous system. These are the sensory and motor nerves. The sensory nerves bring information in. The motor nerves send information out. Now, the motor system can be divided into the somatic and the autonomic branches. The somatic branch will take care of conscious movements. The autonomic system will take care of unconscious movements. And the autonomic system is further divided into the sympathetic system and the parasympathetic system. The sympathetic system is your emergency system. The parasympathetic system is your day-to-day housekeeping system. So let's look at our nervous tissue. The neuroglia are the supporting cells, and there are 6 different kinds of neuroglia. 4 of them are found in the central nervous system, and 2 are found in the peripheral nervous system. In the central nervous system, we have the astrocytes. These are going to anchor the neurons to the blood supply. Also, they're going to act as a barrier between the blood and the neurons. They're going to protect the neurons from some of the substances that may be in the blood. The second glial cell found in the central nervous system is the microglia. The microglia act as phagocytes. They're sort of the immune system of the nervous system. They help dispose of cellular debris and any bacteria that may get in. The third type of glial cell found in the central nervous system is the ependymal cells. These cells line cavities of the brain and the spinal cord. The cerebrospinal fluid will be in these cavities, and the cilia on these cells help circulate the cerebrospinal fluid. And finally, we have the oligodendrocytes. These are the insulating cells. They send processes out to wrap around the nerve fibers, the axons of the neurons, and form the myelin sheaths. The myelin sheaths help keep your wires from getting crossed. They literally are the insulation around those electrical nerves. Here we see the cells. The astrocytes are anchoring the neurons to the blood vessels, and also they're forming this barrier. So they're another layer of material from the blood has to go through to get to the neuron. The microglial cells, these phagocytic cells that can wander around and eat up cellular debris. The ependymal cells, lining cavities with the cilia that help keep the fluid moving in the cavity. And the oligodendrocytes. Notice how one cell sends out several processes to wrap several nerve fibers. This is the insulation for the nerves. All of these are found in the central nervous system. There are only 2 glial cells found in the peripheral nervous system. These are the Schwann cells, and the Schwann cells form the myelin sheaths in the peripheral nervous system. Several cells, however, are involved in making the myelin sheath. Instead of one wrapping many, we have many wrapping one. And then we have the satellite cells, and the satellite cells will protect and cushion the cell body of the neuron. The myelin sheath made by the Schwann cells is made because the Schwann cell wraps several layers around each nerve fiber. This makes what we call the myelin sheath, with the cellular part on the very outside, and that's known as the neurolemma. Besides the glial cells, the other cell of nervous tissue is the neuron. The neuron has a cell body. The cell body is the mass of the cell. Most of the organelles, most of the cytoplasm, and the nucleus are found in the cell body. And then there are processes that come off of the cell body. The dendrites are short branches that come off of the cell body that help deliver messages to the cell body. They pick up information. The axons are going to take messages away from the cell body. Typically, there is only one axon per neuron, but there are many dendrites per neuron. The axon hillock is where the axon is attached to the cell body. The axon terminals are the branching ends of the axon. This is what is going to communicate with the next cell. The axon terminals contain the neurotransmitters. These are the chemicals that transmit information from the neuron to the other cells of the body. When a nerve impulse gets all the way to the axon terminals, it causes the release of the neurotransmitter. Myelin is that fatty covering of the axons. We called it the myelin sheath. This protects the axon, insulates it, and increases the speed of impulse transmission. In multiple sclerosis, the immune system attacks and destroys the myelin sheath of these nerves. This means that the rate of transmission slows in these nerves, and also the nerves stop functioning as well as they're supposed to. The Schwann cells don't completely cover the axon. There are little gaps between the Schwann cells. These are known as the nodes of Ranvier. This allows the signal to travel faster. The signal can actually jump from node to node, making the signal travel much faster than if it had to go the entire length of the axon. The synaptic cleft, you'll remember, is that gap that separates the neuron from its effector cell. We saw this with the muscle cells. When we talk about nervous tissue, we frequently talk about white matter and gray matter. White matter are the myelinated fibers. These are going to be the axons. Gray matter are going to be any of the unmyelinated parts. These are the cell bodies and the dendrites. So as we look at our neuron, here are the many short branching dendrites. This is going to pick up information. And here's the cell body. Now there is cytoplasm and some organelles in the dendrites and in the axon, but the majority of them are found here in the cell body. This is the axon hillock. This is the place where the axon is attached to the cell body. And here is the axon. Notice how the axon is covered with Schwann cells. This is its insulation. But that there are gaps between the Schwann cells. So those are your nodes of Ranvier. At the very end of the axon are the branching axon terminals. This is what will communicate with the next cell. There will be a synaptic cleft between the axon terminal and the next cell. And it is the axon terminals that will release the neurotransmitter. The neurotransmitter is how we're going to tell the next cell what the message is. This is a microscope slide showing as much of a neuron as we can show. We can see the branches that are the dendrites coming off. Here is the cell body with the nucleus. This is the axon hillock, and this would be the axon going off to communicate with another cell. Neurons have two unique characteristics. First of all, they're irritable. That is, they can respond to stimuli. And secondly, they're conductive. They can transmit that information to another cell. A stimulus is any kind of disruption in the area. It could be light, sound, pressure, or vibration. This is detected by the dendrites. The dendrites may pick up this information themselves, or they may be closely associated with some sort of receptor that picks up this information. If the stimulus is strong enough, the message will get to the axon hillock. Now, if the message gets to the axon hillock, an action potential will develop on the axon. This is necessary to transmit the signal to the next cell. An action potential is a brief reversal of the charge across the membrane. We're going to have charged particles moving across the membrane. That's basically electricity. When this gets to the axon terminals, it will cause the release of the neurotransmitter. So let's look at how a nerve impulse is generated. When the cell is at rest, we say it's polarized, it's resting. The plasma membrane is inactive, it's not doing anything, it's not sending any kind of impulse. The ions are arranged on the cell membrane so that we have a collection of positive ions right on the outside and a collection of negative ions right on the inside. The cell spends energy pumping sodium out, and so we tend to have sodium kind of pooled here. It pumps potassium in, but we pump more sodium out than we pump potassium in, so we have sort of a collection of positive ions right here. Now, negatives are always trying to find positives, so the negative ions of the cell sort of snuggle up against the underside of the cell membrane because it has all of these positives on the outside. There are just as many positive ions outside the cell as inside, but the cell makes sure that there's more sodium outside the cell and more potassium inside the cell. This is what we call the resting membrane potential. When the cell is depolarized, the stimulus causes sodium gates on the cell membrane to open. Because there's very little sodium inside the cell, sodium is naturally going to rush in. And sodium is a charged particle, so as it rushes in, it's going to bring those positive ions to the inside. We're going to leave it more negative on the outside, and we're going to reverse the polarity. We're going to make it more positive on the inside, more negative on the outside. The action potential or the nerve impulse happens because this influx of sodium ions here opens another sodium gate here and here and here, and so we have this wave of depolarization go down the axon until we get to the axon terminals. Repolarization occurs because right after we open those sodium gates, we open some potassium gates, and potassium rushes out of the cell. Potassium is positively charged, and so we restore that positive on the outside, negative on the inside charge that we had prior to the depolarization. The membrane goes back to being polarized, but things are not quite normal because now we've got potassium outside the cell and sodium inside the cell, and we have to get that fixed. And until we fix that, we cannot conduct another nerve impulse. The sodium-potassium pumps reset the ions. That is, it pumps the sodiums out of the cell and the potassiums back into the cell, so we go back to our resting membrane potential. It takes energy to do this. Your nervous tissue uses a vast quantity of energy, and if you don't have enough ATP, your nerve will actually be unable to repolarize. This is the axon coming down to an axon terminal, and when we get that action potential all the way down here, we're going to open calcium gates, and when calcium comes into the cell, that causes the release of the neurotransmitter into the synaptic cleft. Now this neurotransmitter will attach to receptors on the receiving cell. This could be a muscle cell, another nerve cell. Whenever the neurotransmitter attaches to these receptors, there is going to be a change on the cell membrane of this cell. Remember we saw with muscle cells, this is what caused the action potential to occur on the muscle cell. Myelinated fibers have impulses that travel much more quickly. The impulses don't have to travel along the entire axon membrane because of the myelin sheath. Rather, it jumps from node of Ranvier to node of Ranvier. This is called saltatory conduction. This means that the ions are concentrated at those nodes. Only a few have to move in and out of the membrane, so it's quicker to reset the membrane. We also need less energy because we have fewer pumps that have to operate to reset the membrane. Sedatives and anesthetics block nerve impulses, usually by blocking the opening of sodium gates. They alter the membrane permeability so the charges don't reverse. Cold and pressure can also block nerve impulses. This has to do with a lack of oxygen and nutrients from the blood vessels getting there, so we don't have enough energy to reset the membrane fast enough. Once the impulse reaches the axon terminals, the neurotransmitter is released via exocytosis into the synaptic cleft. The neurotransmitters will bind to receptors on the next cell and cause that cell to depolarize. This whole process is what we call an electrochemical event. It's electrical because we have an impulse traveling to the end of the axon. It's chemical because that electrical impulse causes the release of the chemical neurotransmitters. The central nervous system is composed of the brain and the spinal cord. The brain is divided into four major regions. We have the cerebrum, which is the largest part of the brain, the diencephalon, which is located pretty much here in the center of the brain, the brain stem, which is attached just to the spinal cord, and then the cerebellum, which is tucked up under the brain. The cerebrum is the most superior part of the brain, and it's the largest region of the brain. It's divided into left and right hemispheres with 5 lobes on each side. When we look at the cerebrum, you'll see elevated ridges covering the surface. These are known as gyri. A singular is gyrus. The grooves between the gyri are sulci. A singular is sulcus. There are some deeper grooves that separate large regions of the brain. These are known as fissures. The cerebrum is your conscious brain, so it controls your conscious activities: speech, voluntary movement, memory, logical and emotional response, consciousness, and this is also the part of your brain that interprets sensations. You know that you're cold, you know that you're seeing the color red, those kinds of things. The diencephalon can be divided into 3 regions: the thalamus, the hypothalamus, and the epithalamus. This is part of your unconscious brain. Things that come into the diencephalon are directed to the higher brain. So input that's coming in, if it needs to go to your conscious brain, it's directed there. If it needs to go to parts of your unconscious brain, it's directed to the unconscious part. Your diencephalon houses controls for body temperature, water balance, and metabolism. This is also where your emotional center is housed. Your food, sex drive, pain, pleasure drives are all located in the diencephalon. The hypothalamus, that small part of the diencephalon, is your major homeostatic center. The brainstem extends downward from the brain. It too has three regions, the midbrain, the pons, and the medulla oblongata. This controls very basic life functions: breathing, blood pressure, reflexes, heart rate, swallowing, and vomiting. If this part of the brain is damaged, a coma can result. The cerebellum is tucked under the cerebrum and behind the brainstem. It too has left and right hemispheres. This is an unconscious part of the brain, but it helps control the timing of muscle actions and your coordination. Whenever you go to move your arm, that's a conscious thought that you're going to pick up a book, for example. But dozens of muscles are involved in that movement. It's the cerebellum that makes sure all of the muscles move at just the right time so you have a nice, smooth movement. If the cerebellum is damaged, you may have something called ataxia. This is clumsy movement. Head trauma may cause brain injuries. The brain may actually bounce around inside the skull and do damage to the brain. A concussion is considered a slight injury. People may or may not lose consciousness as the result of a concussion. One concussion probably doesn't hurt you too much, but studies are being done to make sure that repeated concussions do not cause permanent brain damage. With a contusion, the tissue bounced so hard against the skull that there was actually some tissue destruction. The brain is bruised. This can lead to coma. Intracranial hemorrhage or edema occurs and the brain may swell. This can compress the brain tissue, and when that happens, blood flow is blocked. This means oxygen cannot get to the neurons. Neurons, remember, need a lot of ATP to function properly, and they need oxygen to make that ATP. If they fail to get oxygen, these neurons die, and neurons cannot replace themselves. Strokes, or cerebrovascular accidents, or CVAs, occur when blood circulation is blocked. This could be as a result of a clot in a blood vessel, a ruptured blood vessel, swelling of the brain, or tumors that put pressure on blood vessels. Again, the part of the brain that does not get blood will die. This means that this part of the brain will become non-functional.